Dr. Stephen Rader is a professor of biochemistry and molecular biology at UNBC. Dr. Rader has many published articles on topics ranging from pre-mRNA splicing to hydrogen bonds and sulfur atoms and proteins. Welcome, Dr. Rader. Let's get to know your spicy mind. How would you like me to address you? I am Sharon, and I use she, her pronouns.
And I am Stephen, and I use he, him.
Thank you. We'll start off easy. Where did you grow up? Can you tell me about your hometown?
Yeah, it's not a simple answer. So I was born in San Francisco, and I lived for 3 years outside of Paris. That's sort of my earliest memories. But I grew up mostly in Urbana, Illinois, so about 150 kilometers south of Chicago.
Oh, wow. Tell me what it was like growing up there.
Yeah, actually, it was a wonderful place to grow up. It was one of those places that's small enough that all of your friends hung out in the neighborhood. So in the summer, we would have neighborhood games of capture the flag and things like that. We'd just be running all over through the alleys and through everybody's yards. It was a really nice neighborhood to grow up in. That was great.
Oh, that sounds idyllic. Okay, are you ready for your first hot sauce?
I think so, as
Okay.
ready as I will ever be.
The first sauce is a peanutty sauce at the far end there, yes.
Okay.
It's got a little bit of heat, but not too much. So now that you have peanut butter mouth, because quite a thick sauce, can you please tell me about the 3 most influential people in your life and how they have impacted you?
My mother is probably number 1. And her family is from Hungary. She was born in Hungary. She grew up in England and Scotland. And she studied math and economics at university and has, sort of a classical education. She read a lot of classic literature and studied some Latin and Greek. And she really transmitted those values to me, the sort of values of a liberal education, really trying to know lots of things. When I was growing up, she was always singing, and she had this sort of bizarre repertoire of Gilbert and Sullivan operettas and, university camp songs, like, they would get together at the pub on a Friday and sing these songs together. So I would say she's the sort of main influence on a lot of the ways that I see the world and some of the things that I wanted to do. I would say my second major influence was I was an undergraduate and I was taking a biochemistry course. And the professor who was teaching that was a woman named Judy Vogt. She's quite well known. She wrote one of the textbooks, one of the widely used textbooks in biochemistry. And one of the topics that we studied in that course is something called structural biochemistry, or more specifically X-ray crystallography. And I was so fascinated with that I decided I had to study that. And so that really sort of set the course of my career and scientific development, having been exposed to that. The third strongest influence in my life, I would have to say, is my wife, Martha, who runs the lab at UNBC with me. So we run the lab together, and we have 2 children together, and she, one of the earliest things was getting me interested in music. She played the flute, and I had never really played anything. So I learned to play the flute with her. And she's not only my life partner, we do all sorts of skiing and backpacking and various adventures together, but she also, as I said, we run the lab together, and she's sort of my scientific and career partner.
So it's time for hot sauce number 2.
Can I have a drink of milk?
Yes, you certainly can. So this next sauce is still very mild. It's very thick, almost like a squash soup sort of texture, and it's got a lot of root vegetables in it.
Oh, wow. Amazed that you said thick after the peanut butter one. I was thinking, what could be thicker than that?
Okay, so it's time for me to get a little bit more existential with you. If you could go back and give your 16-year-old self one piece of advice, what would it be?
Ooh. Can I give 2 answers?
Sure.
Okay, so my less serious answer is that I would have, I think my 16-year-old, I was so square growing up. I never tried anything, and well, I won't go into the details, but let's say later in life, I have had the opportunity to experience some of those things, and it's like, maybe I should have tried this sooner. But I would say more seriously, looking back over my life, and especially as a 16-year-old, I took a lot of things for granted. And a lot of opportunities came my way, not necessarily because I deserved them, but just because of being in the family that I was in, that, you know, both of my parents had been to university, and they just had certain expectations, and I just figured things would work out that way, and they just kind of did. And I think I would encourage my younger self to think more, you know, to look more broadly at the world and think about what the opportunities were. I never really thought about, you know, what is my role here? What could I be doing to make the world a better place? I just sort of took things for granted as they came, and I think I might have done some things differently had I known then what I know now.
Okay, thank you. Okay, so when you are ready, we're going to go on to sauce number 3. Sauce number 3 has gold leaf in it.
Gold leaf, wow.
There is gold leaf in it.
You guys didn't hold back for this.
No, I like to think of it as a slightly spicy hollandaise sort of flavor for this sauce. It just has a little kick in there.
I really like that one. It's got a very nice balance.
Okay, so now, still being a little bit existential, if you were a color right now, in this moment, what color would you be and why?
Well, definitely purple. Purple's my favorite color.
Okay.
And it always has been. And I think, I don't know, it just makes me feel happy. It's a happy color.
Okay. Very clear, concise answer there. Are you ready for another sauce?
I am.
So the next one is our first green sauce of the evening. This was a jalapeno cilantro.
That sounds good.
So it's definitely the runniest sauce so far. So it's time for our rapid fire question, but I'll let you swallow first.
I appreciate that.
So for the rapid fire questions, please answer as fast as you can with one-word answers, if you can. Okay, so first of all, you ready? What is your pet peeve?
People park, no, just one word, parking.
Parking, okay. What is your favorite word?
Oddment.
Okay. What is your least favorite word?
Scrotum.
Could you name a 4-letter word that starts with the letter B?
Bomb.
If you had to change your first name, what would you change it to?
Alexander.
What is your favorite pet?
Snake.
Have you ever worn socks with sandals?
Yes.
Thank you. You'd be surprised how many people try to not admit that. Okay.
I think as a university professor, we actually embrace that.
Okay, so sauce number 5. So we're right here in the middle. This one is a very interesting flavor. It's a habanero curry with a lot of orange, but it's quite a sweet sauce.
That's really nice.
What are your favorite things about being in northern BC?
I often tell people that the Otway Ski Center is the jewel of Prince George. It's such an amazing facility. I ski there 3 or 4 times a week, the whole season that it's open. And you know, being able to go cross-country skiing before work, how many places in the world can you do that, right? It's, you know, probably in Scandinavia they do that, and that's maybe the only other place in the world. So I feel we're just super lucky to have that here in Prince George. And in general, all of the outdoor opportunities here. You know, we do quite a bit of backpacking in the winter, and backcountry skiing. And in fact, that's a big part of the reason that we moved up here, was for the outdoor opportunities. We did a fair amount of that in California before we moved up, but you know, there you had to drive a lot further, and here everything's relatively close. So that's a huge part of it. Another thing that I love about Prince George is that it's small enough that you feel like you're part of a community. And every time, you know, we love visiting Vancouver, we love going to the restaurants there. Obviously there's much bigger variety than there is here in Prince George. But then, you know, you get in traffic and people are honking at each other, and you're sitting for what seems like hours at traffic lights waiting, and you come back here and nobody ever honks. You know, if somebody honks, everybody looks at them and like, what's wrong with you? And you know, you never wait at traffic lights. It's just a really nicely sized community, and I think that makes people more friendly and more supportive. Anywhere you go, you run into people you know, and the people you don't know are friendly, and they're happy to chat, and so I think there's a lot of really great things about the community.
Sauce number 6. This will be our second green sauce. This one is a local sauce made here in Prince George.
Yeah, these are great. I was expecting, as you say, that they would all just be hot, but actually they all have really nice flavors.
Tell me about your field of study.
Okay. So I study a biochemical process called pre-mRNA splicing. And you may have heard that, well, so first of all, we've all been going through the pandemic, of course, and so you've undoubtedly heard of RNA vaccines. So that's the same kind of RNA that we study. And we study something that happens to it in our cells. So you may have heard that DNA is the blueprint of the cell. DNA is what we pass on, you know, to our children and their children. It has the instructions for making everything in our bodies, basically. And the primary instructions that it has are for making proteins. And we know a little bit about proteins. We know proteins are in our diets. It's important to get enough protein in your diet. But it turns out that proteins are individually fascinating. They do all of the things in our bodies. Some of them digest our food. Some of them make our muscles work. Some of them make our neurons in our brains carry electrical charges. And each one of those, the instructions to make it are in the DNA. And there's a bit of a logistical problem that the DNA, we just basically have one copy of the DNA, and it's in the center of our cells in something called the nucleus. And there are thousands of factories in our cells that make the proteins. So how do you get the one instruction to all of those factories? They all need a copy of the blueprint. And so it turns out that messenger RNA is exactly those basically photocopies of the blueprint. And there's one additional little wrinkle to this story, that the instructions that are in our DNA have interruptions in them. It's as if you had a multi-page blueprint for making, you know, a complicated building, say, and in the middle there were pages with ads on them. You're like, why are there these ads in the middle of my blueprint? And so in order to make the building, you would have to rip out the ads. That's exactly what our cells do with the messenger RNA. So after it gets copied, after the instructions get copied from the DNA into the RNA, the cells have to go through and find the interrupting bits and cut them out.
Okay.
And it turns out that there are lots and lots of diseases that are caused by problems in this process. So this process is called splicing. It's called RNA splicing. And if there's an error in it, because almost every gene in our bodies has these interrupting bits. And so if they're not spliced out properly, it causes all sorts of problems. And so cystic fibrosis, for example, various kinds of cancer, spinal muscular atrophy, all sorts of diseases are caused by these problems with splicing. And so we and many other scientists around the world are interested in understanding better how this process works. And so we actually study it in a red alga, which is found in volcanic hot springs. And the reason we picked this alga to study is that in our bodies, we have about 200,000 of these interrupting bits in the genes. And it's really hard to do experiments, you know, to sort of look at all of them and understand what they're doing. But this alga only has 38. It has the smallest number of any known free-living organism, anything that's sort of living on its own. And so we decided to study it there because it's, first of all, it's easy to manipulate. You can grow lots of them in the lab. And nobody minds if you do experiments on algae.
Yeah.
And so then we can try to test each one of these interrupting bits and say, what is it doing? Why is it still there? We know that the ancestor of this alga had about 2,000 of these. And it's lost almost all of them. But it's still retained 38. And in fact, if we look across all species where people have looked at this, it's very common that they've lost these interrupting bits over time. But they've never eliminated them. So somehow there are always a few that hang on. And we suspect that there's probably some kind of conserved thing that they do, some important thing that they do that's common to all of these organisms that makes it so that they can't completely get rid of them. And it would be much easier if they could get rid of them because it takes energy to keep them in the cells and to process them and do everything that has to happen. And it gives more opportunity for errors to arise as well. So certainly microscopic things like this alga that we study would probably be better off without them. And so that's actually one of the things that we're trying to do is to take them all out of the genome and see, does it grow better than the normal strains? Does it have any effect?
Oh, that's really interesting. That's really neat. Are you ready for another sauce?
I am ready.
Okay, so we are up to number 7, our first real orange sauce.
Okay.
And this one is sort of the most traditional flavored hot sauce. And this one is also made locally here in Prince George. In today's world, why do you feel that your field of study is important?
Well, I hope that the pandemic has brought home the importance of ribonucleic acid, or RNA, to everybody. And so the field itself, we know sort of, in sort of the popular consciousness from the pandemic, we know sort of two sides of it. First of all, the virus is an RNA virus. And so that points to the fact that RNA has a lot of interesting roles in life, and that knowing more about RNA is probably a useful thing. The coronavirus that infected us is definitely not the only RNA virus. There are lots of other ones out there, and so studying them and understanding how they work is going to be very important. And then the RNA vaccine is the other big thing that people know about, is that, oh, hey, here's this technology that nobody had heard of until 2 years ago. And it suddenly, sort of broke on the scene and came to the rescue and, really helped us change the course of the pandemic in sort of record time. And what that points to is this whole area of molecular biotechnology. So there are lots and lots of companies out there that are looking at how can we improve healthcare, how can we diagnose diseases more effectively, how can we generate products that will be useful and help society. And how can we do that in a renewable and sustainable way? And so RNA is at the center of a lot of those things. There are lots and lots of companies right now. So Moderna is now famous, for example, for making this vaccine. But actually, they didn't even think about vaccines when they got started. They had a lot of other diseases that they thought they might be able to cure with this technology. And so both the sort of fundamental science side of it, what is RNA doing in our bodies? How does it work? What are all the processes that it's involved in? And then also the technology side. How can we use it to cure diseases and to make interesting products? I think both of those things really sort of capture why it's important to be studying RNA.
Are you ready for number 8?
I am ready.
So number 8 has a very scary bottle, but I have to use a fermented ghost pepper for its heat.
Fermented ghost pepper, wow.
Yes, and again, this one is also made here in Prince George. What is one lesson your field of study has taught you that you think everyone should learn at some point in their life?
And it's not really specific to my field of study, but to science in general. It's the importance of trying to unlearn our habits of jumping to conclusion and instead looking at the evidence and thinking about what's actually happening here. What does the evidence tell me is really going on? And this is something, of course, that we do on a personal level all the time with our friends, where, you see your best friend talking to your boyfriend and you're like, what's going on there? And you just jump to conclusions instead of, trying to get more information about it. And obviously that's a fairly trivial example, unless you're in that situation. But there's all sorts of things that go on in society, mask wearing and the COVID mandates and all of those things obviously riled up a lot of people. And it's really difficult because there's sort of a clash then of, what are my personal values? What do I think is important? How is this impacting me? But then what is the effort? What is the public policy trying to do? And, can I think about the overcrowded hospitals with patients spilling out onto the streets and say, okay, if I change my behavior, that will make a difference. And so it's really that sort of taking an evidence-based or sort of scientific way to look at the world, I think is the biggest thing that I've taken away from being a scientist, not specifically from studying RNA.
To number 9, this hot sauce comes from the Okanagan and is more of a barbecue sauce. It's a very flavorful.
It's also delicious.
What do you consider your greatest professional achievement? It's a hard question, I know.
It is a hard question. And I'm thinking on the one hand about the students I've trained and the impact that I've had helping young people to figure out what they want to do and helping them get to the positions that they have. And on the other hand, the things that we've discovered scientifically in my lab. I'm going to go with the scientific discovery. And it's related to what I was telling you about how this alga that we're studying has only 38 of these interrupting regions. And a few years ago, we published a paper where we looked, so on the one hand, you have these interrupting regions in the genes. And in order to remove them, in order to splice them out of the messenger RNA, there's a whole complicated machinery. In humans, we have about 250 proteins and 5 other little RNA molecules, and they all have to come together in the right way and recognize these bits and pull them out. And we discovered that in this alga that we're studying, that has so few of these interrupting regions, its machinery is much less complex. So it only has about 45 of these proteins, and it's missing an entire particle that is normally the first step of recognizing this interrupting region. And it's the first organism that's been found to be missing this whole sort of subset of the splicing machinery. And so, yeah, to us, that was a pretty big deal. And we're very excited to find that. And it was done, with UNBC students, including undergrads who were helping out on that project.
Now it's time for our last sauce, if you're ready.
I'm ready.
This is the sauce that has the most heat today, and it has pineapple in it. So now that you have tasted our gauntlet of sauces from all over beautiful British Columbia, we have sauces from Vancouver Island, we have sauces from Tofino, from the Okanagan, and several from right here in Prince George. It's time for you to let us know anything that you would like our viewers to know. For example, what's next for you? Any special projects coming up, or just a shameless self-plug?
Well, we're continuing our RNA research. As I mentioned, one of the things that we would like to do is to remove all of these interrupting bits from this alga, which would make it the first organism where this whole system has been taken out, so we can see what happens. So we have a couple of students in the lab who are working on that right now. It takes a while to do each one, so we think it will probably be around 3 years before we're finished with that, if it turns out to be possible. Another really interesting thing that we've started doing is we have a couple of collaborators, one of them in particular in Saudi Arabia, who is interested in using this same alga that we study for biotechnological purposes. Well, this is the first one that has actually been...
And it grows on you. You think you're good, and then it just sort of blooms, right?
That's right.
That's why you've got the milk.
Yeah. Exciting things about algae for sort of biotechnology purposes is that they basically eat sunlight. So they get their energy from sunlight and from CO2. So they also are taking CO2 from the atmosphere. And so it's a very sustainable way to produce things. And one of the challenges is that the media that you grow them in can be quite expensive, and you have to grow them in special containers. The alga that we study is an extremophile. It lives at high temperatures and acidic conditions. And so it lives under conditions where it's not easy to contaminate it with other things. So we can grow it in open pools. And this colleague has shown that he can grow it in the desert in Saudi Arabia in seawater.
Oh, wow.
Which of course is very cheap. And so we're hoping that there will be some interesting things that come out of that in terms of using algae to make cool things. And you know, one of the things that I would love to do is, for example, to team up with some of the local industries, because they're emitting hot gases full of CO2. And so if we can bubble that through algal cultures, that would heat them up and also provide the CO2 that the algae need to eat. So those are some of the things that we're looking at in terms of the work in the lab. On a personal note, I have been working on a play.
Ooh.
And so I took part, there was a workshop through Theatre Northwest, and I took part in that. And I'm hoping that, I haven't quite finished writing it, but I'm hoping that in the next few months I'll get that finished and that we can actually try producing it somewhere.
Oh, that would be fun. That is very exciting. Thank you so much for coming in and enjoying all of these hot sauces with us.
Well,
it's been
thank you.
fascinating.
Thank you. It's been a pleasure.